In this video, we will discuss about some of the in vivo results of this GlyTR1 and GlyTR2 in the xenogeneic humanized mouse, and they have checked different cancers, different types of cancers. So in the panel A, they have shown the immunofluorescence image of the triple negative breast cancer with D-luciferin, that is luminescence. Here we can see the general tumor image, and the GlyTR1 detections or fluorescence is also can be seen in this panel A. And similarly, they also did the similar things with GlyTR2, and we can see that GlyTR2 uh, fluorescence can also be seen in the site of tumors for this triple negative breast cancer. So this is just to show that the GlyTR1 and GlyTR2 also has a fluorescence, and it is usually found in the tumors. Thereafter, they checked it first about this pancreatic cancers, where they used Capen-1 tumors that are induced into the mice, and then for the control they induce this PBS. And what they are able to see, that in the case of PBS they are able to see a growth in the tumors, whereas in the case of GlyTR1 induced CAR, they are able to see a regression of the tumors, and the quantifications are shown in this panel D and panel E. For panel D is about the pancreatic cancers. Here we can see that the total flux of the, or we can say the tumors formations, is increased in the case where GlyTR1 is not been used. And this is the point, that is around at day 4 the CD8 plus T cells are induced, and we can see that a decrease in the total flux for the case where GlyTR1 containing CAR T cells are induced into the mice. And similar results they have seen for triple negative breast cancer cells, where we can see that in the case of control they are able to see a sustained tumor growth, whereas in the case of GlyTR1 they are able to see a regressions in the tumor growth. And these things are also quantified in the panel E, where they have shown the results of control, where the total flux is continued to increase, whereas for the GlyTR2 the flux is lower than the control, and for the GlyTR1 the flux is even lower compared to the controls. And when they check it for this, the panel F is about the triple negative breast cancer with GlyTR1 CAR T, and panel G is about triple negative breast cancer with GlyTR2 CAR T. And here we have seen some tumors compared to the case of some high tumors in the GlyTR2 compared to the case of GlyTR1 in the triple negative breast cancers. And however, when we see the quantifications, we can see that the overall flux is higher in the control compared to the GlyTR1 and GlyTR2, and the p values are lower than the 0.05 for the GlyTR1 also. And when they check it for this SCOV3 or the ovarian cancer, they are able to see a decrease in the or regressions in the tumors for both GlyTR1 and GlyTR2. And these things they have quantified in the panel I, where we can see that a total flux is low and statistically significant compared to the control for both GlyTR1 and GlyTR2, indicating that their GlyTR1 and GlyTR2 has a role towards the regression of the tumors. And the panel H, they have shown the results for this triple negative breast cancers, but in this case it is kind of like mutated, where I'm not able to recall what this C represents, but the results are similar. That is when they use different concentrations of this GlyTR2, they are able to see a decrease in the total flux as the concentrations of this GlyTR2 increases, indicating that GlyTR2 is also working in the triple negative breast cancers. However, its dosage requirements seems to be slightly higher compared to the case of GlyTR1. So I hope you understood how they validate their CAR GlyTR1 and CAR GlyTR2 reconstruct on the in vivo xenogenic humanized mouse. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.